Hello, everyone. I'm Pia Anderson here with Elise Lamont, and we are here to present to you the fascinating structure-function relationship of the Zika virus capsid protein. Our thesis statement, as depicted on the screen, reads as follows. The alpha-5 helices of the Zika virus capsid are necessary for the connectivity of multiple capsid dimers to each other to create the quaternary structure as well as promote their interactions with the transmembrane regions of the surface PRM and E proteins that are vital to formation of new viral particles. Now, that was a mouthful. So before delving into the intricacies of the Zika virus biochemistry, it's important first to take a look and reflect on why this is an important biomolecule to discuss. So what makes the Zika virus relevant today in the midst of a brand new and fear-provoking coronavirus pandemic? Well, to put it curtly, for many individuals, the Zika virus disease is more than just its usual symptoms of headaches, itchiness, and fevers. It's a dangerous condition that can cause severe neurological complications, both to infected patients and to the fetuses of infected women. Oftentimes, however, infected women don't even make it to full-term pregnancy, having miscarriages or preterm birth. This image to the left shows a sketch of a newborn suffering from microcephaly, a congenital condition often caused by Zika infection during pregnancy. This is a severely debilitating disease, physically, intellectually, and emotionally, characterized by a shrunken head and below average intelligence. The image to the right shows a young woman suffering from Guillain-Barré syndrome, a rare disorder in which the body's immune system attacks the nerves. Symptoms begin as mere weaknesses and tingling in the extremities, but can quickly spread to full body paralysis. As of 2016, CDC research suggests a strong association between Zika virus and an increased risk of developing Guillain-Barré syndrome or other neurological disorders.
Before we delve into the specifics of the Zika virus structure and function, it's important that we cover some relevant terms. Zika virus is a flavivirus, which means it's of the Flaviviridae family. They are plus-sense, single-stranded enveloped RNA viruses, found in arthropods, primarily ticks and mosquitoes, and can occasionally infect humans. Next is a capsid, which is the protein that encapsulates the viral RNA or DNA. It can have a polyhedral, helical, spherical, or complex structure. Lastly is the envelope, which surrounds the viral capsid and serves to avoid immune system detection and identifies and binds to receptor sites on host cell membranes. It's also important to cover the route of infection for an enveloped virus such as Zika virus. First, the coated pits of the host cell coat the virus via envelope proteins. Viruses are tissue-specific due to specific receptors, since certain tissues only recognize certain proteins. Often these proteins are within the envelope of the virus. The envelope proteins allow the virus to bind to the receptor. So the enveloped virus binds to receptor and then enters the cell as a vesicle. The vesicle then turns into an endosome, which moves towards the ER membrane. The pH goes down in the endosome, which releases the capsidized virus, resulting in delivery of the virus to the ER membrane, where it can begin replicating using host cell machinery. Shown on the left is a photo of a viral capsid, precursor membrane, and envelope. The capsid's essential roles are protection of RNA when the virion is outside of a host cell, and facilitation of replication when the virion contacts a receptor on an appropriate host cell. So, changes in the capsid cause or accompany loss of viral infectivity, rendering the virus ineffective.
These three proteins, C, PRM, and E, that Elise just mentioned, are structural proteins used to construct the new virus shells. E and PRM come together to form E-PRM complexes. Each capsid protein dimer binds to 3 of these E-PRM protein complexes, creating a triangular network of capsid protein dimers. Without the capsid protein, that complex is rendered ineffective and produces irregular, sometimes non-functioning, new virion shells. This highlights the importance of the capsid protein, a monomer of which is shown on the right. Next, we'll get a more up-close look at the structural features of the capsid itself that will contribute to its function. The alpha-5 helices on the capsid protein play an essential role in the trimerization of capsid dimers. In other words, the hydrophobic interactions that take place on and around the alpha-5 helix allow for the formation of the protein capsid's quaternary structure, which, along with the structural proteins, form the previously mentioned assembly unit. As you can see on the left-hand image, the three dimers are brought together in a triangular network through the alpha-5 helix, which is coded in red. The photo on the right represents one dimer present within the trimer. The alpha-5 helix of the capsid protein is often thought of as an extension of the main dimer, thus explaining why it is not depicted in the picture on the right. This can be seen as an accurate representation, because the fifth alpha helix is usually cleaved off at some point during the virion replication. All in all, however, the most important takeaway here is how a viewer can see the role of the fifth alpha helix in bridging together the entire quaternary structure. The hydrophobic interactions on the alpha-5 helices drive the formation of the quaternary structure by breaking the hydration shell and providing the energy needed to create the trimer. The left portion of the image here on the slide gives an overview of the Zika virus capsid fit in the context of the virus's quaternary structure. However, the key structural feature, the alpha-5 helices, are enlarged to the right. Here you can see how the two capsid dimers interact via hydrophobic residues that come together, making a greater triangular network. To expand just a little bit more on the hydrophobicity of the alpha-5 helices, we should zoom in and observe the amino acid sequence of this fifth alpha helix. While this chart may show the amino acid sequences of several flaviviruses, we're only really concerned with the top row, which shows that of the Zika virus. As you can see, the image depicts from residues 103 to 115 the amino acids that compose the alpha-5 helix. Upon closer inspection, you can see that of these 12 amino acids, 9 are nonpolar amino acids, most notably the 3 leucines on residues 110 through 112, along with valine, glycine, and isoleucine. This just reiterates the fact that the hydrophobic chemical properties of this structural unit within the capsid contribute to the overall formation of the biomolecule.
In Zika, there are two main functions of the capsid protein, which are structural integrity of the virus to provide protection of the viral genome by tight-knit trimerization of capsid proteins. The capsid is also responsible for communicating with other components of the viral polyprotein. Both of these highlight the alpha-5 helix as being necessary to carry out either function, as I will explain in the next slides. The fifth alpha helix is essential to quaternary structure of the viral capsid because, as Pia said, the fifth alpha helix allows for formation of trimers of capsid protein protomers. Residue R32 in the alpha-5 helix of one protomer forms 2 hydrogen bonds with the R32 of another protomer. Residues P26 and G29 in the alpha-5 helix of one protomer make hydrogen bonds with L63 and K60 in the alpha-2 to alpha-3 loop of another protomer, respectively. Thus, the Zika virus capsid protein forms a tight homodimer through extensive contact networks using these hydrogen bond interactions, as well as the hydrophobic interactions Pia explained before. These two together provide the capsid's structural support in its job of encapsulating and protecting the genomic material. Regarding the second function of the capsid protein, as Pia shared, there are 9 hydrophobic residues that are within the 5th alpha helix. These allow it to be threaded back and forth through the ER membrane, unlike other alpha helices of the capsid protein. Because of its cross-membrane conformation, the 5th alpha helix serves to direct translocation of PRM proteins into the ER lumen to communicate with the E protein and form the assembly complex we discussed earlier. The mechanism by which the alpha 5 helix serves as a signal peptide to do such translocation has not been thoroughly explained in published literature. However, viral assembly involving only E and PRM proteins is unable to produce Zika icosahedral particles, so we know that the capsid's role in viral replication is necessary. Thus, the alpha 5 helix is vital to perpetuating the cycle of infection because it facilitates communication between the rest of the capsid of the capsid protein that sits outside of the ER membrane and the glycoproteins that are embedded in the ER lumen. In conclusion, the 5th alpha helix on the capsid protein serves as an important communicator between the capsid and the surface glycoproteins E and PRM, as well as serves to ensure that the virus is packaged properly to protect viral RNA. You want to pause it?